It was the first human evidence that something other than genes passed between generations. Something that could control genes directly, switch them on or off. But how exactly did these tags go about silencing a gene? This odd strain of agouti mice provides a visual clue. Despite the difference in color and size, they're twins, genetically identical. Both, therefore, have a particular gene called agouti, but in the yellow mouse, it's switched on all the time.
And as a consequence, it inappropriately blocks a receptor in what's called the satiation center of the brain, which tells mice and us when we're full. So the yellow animals literally eat themselves into obesity, diabetes, and cancer.
So what switched the agouti gene off in the thin mouse? Exercise? Atkins? No. A chemical tag called a methyl molecule. Composed of carbon and hydrogen, it affixes near the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl molecules, attach to DNA directly. Other types grab the proteins called histones around which DNA wraps and tighten or loosen them to turn genes on or off.
And in simple terms, this contact can be thought of as hugging the DNA. And if these proteins hug the DNA very tightly, then it is hidden from view for the cell, and a gene that is hidden cannot be utilized.
These tags and others control gene expression through a vast network in the body called the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, and how much.